This video is the third video in Unit 6, which is Gene Expression Regulation. And this video specifically is going to cover transcription and RNA processing, as well as translation, which are Units 6.3 and 6.4. So to start, let's review what is gene expression. So gene expression is when you have DNA instructions that is then transcribed into mRNA, and then that is translated into a protein. So we're going to be looking at each of these steps in detail in this video. First, let's look a little bit at different cell types, so eukaryotic versus prokaryotic gene expression. In prokaryotes, transcription and translation take place simultaneously, whereas in eukaryotes, transcription and translation take place sequentially. This is just one of many examples of how eukaryotes are a little bit more complex than prokaryotes. Complexity is not always better. Sometimes simplicity has some real advantages. But regardless, gene expression is more complicated in eukaryotes and also includes RNA processing as a step. The process of transcription is that first step of gene expression, and here is where we use DNA as a template to create a strand of RNA. Remember that DNA and RNA are both nucleic acids, which is why the term transcription makes sense, because we're still in the nucleic acid world. We're just kind of copying it. We're rewriting it in a slightly different form, but RNA and DNA are very similar. They're both nucleic acids. They both involve nucleotides. Please note that only one strand of the DNA is used during the transcription of a gene. So take a look at this diagram. Which strand in this case is used as the template strand? The template strand is the strand of DNA that's being read. So in this case, it's the one on the bottom. Another term that's used for distinguishing which DNA strand we're referring to is sense and antisense. Now this is a little bit counterintuitive. Sense and antisense are based on how similarly the DNA strand matches the RNA transcript. So if you look at the transcript down there, the RNA, it's AUG, AUC, UCG, UAA. Which strand of the DNA more closely resembles that strand? And that would be the top strand of the DNA. So this is called the sense strand. And the reason here is that the sense strand and transcript have similar codes, other than the difference between T versus U. And that's because they're both complements of the DNA template strand. So they're both mirror images, essentially, of that DNA template strand. And that's why the non-template strand is called the sense strand, whereas the template strand is sometimes called the antisense strand. So let's go through the three major steps of transcription, which are initiation, elongation, and termination. But before we do this, I just want to make sure you're understanding that this isn't really very complicated. Yes, there are some details, but at least the overall steps are pretty straightforward. What would be other words you could describe these three steps? Initiation is just starting, elongation is growing, and termination is ending. So for the steps of transcription, we start, we grow, we end. Let's look at all of those in order. An important term in transcription is promoter. Take a look at this diagram and describe the function and type of biomolecule of the promoter. So the promoter is nucleic acid. It's a DNA region. So it's the part of the DNA where transcription begins. This region contains a sequence of nucleotides that are recognized by transcription factors that lead to the binding of RNA polymerase. So this is where transcription starts. Next step is elongation. Here we have growth. So here we have a picture of RNA polymerase attached to the DNA. So describe the function and type of biomolecule of the RNA polymerase. RNA polymerase is an enzyme, so it's a type of protein, that builds the mRNA strand using the DNA as a template. So it adds nucleotides to the growing RNA strand, very similarly to what we saw DNA polymerase do, but here we have RNA polymerase. So we're building a polymer. We're building that nucleic acid strand from nucleotides of RNA, using that template DNA as an instruction manual. A couple of questions here. First is, what direction does RNA polymerase read the template DNA? And what direction does RNA polymerase build the mRNA? Just like we saw in DNA, or DNA replication, the polymerase, in this case RNA polymerase, is going to read the template in a 3 to 5 direction, reads 3 to 5, and it builds in the 5 to 3 direction. On this diagram, which way is the RNA polymerase moving? If we know the direction of both reading and building, which way is the RNA polymerase going? In this diagram, the RNA polymerase is moving from left to right. Now, you can't always assume that. In a lot of diagrams, it is going to be shown of transcribing from left to right, but that's not always the case. Please pay attention to the 5' and 3' ends, and remember that we're building in a 5 to 3, reading in a 3 to 5. Why is it that DNA and RNA always build in a 5' to 3' direction? That's because you need that open hydroxyl group, that open OH, to grab onto, and you need that 5' phosphate end of the free nucleotide is what's going to make that connection. So it's always going to be building in a 5' to 3' direction because it's that 3' open end that needs to join onto the free nucleotide. This diagram is just another visual of showing the process of transcription. It's showing that template strand of DNA and the RNA strand being produced. And it's also in the bottom pointing out that there are no T's in the RNA. Instead of T, we have U. The final step of transcription is termination. Remember, this is when it ends. The details of this are beyond the class. There are a couple of different mechanisms that can cause transcription to stop. You don't need to worry about it. Just know that it doesn't go forever. At some point when we reach the end of the gene, the RNA stops building. This diagram is showing a review of all three of the processes of transcription, initiation, elongation, and termination. After transcription is completed, in eukaryotes we have a process called RNA processing. In prokaryotes, this doesn't happen, and the RNA just goes on to translation. But in eukaryotes, the RNA is processed. Take a look at this diagram and figure out if you can find the three ways in which RNA is processed. So two of the changes that happen during RNA processing is the RNA strand gets a cap and a tail. So on the side of the RNA that was first transcribed, the 5' end, it gets a GTP cap. You don't need to know the details of that. And then on the side that was finished, the end of the RNA, it gets a tail, a poly-A tail, because it has a whole bunch of adenines attached to it. Again, you don't need to know the details here, but you should know that the cap and the tail help protect the mRNA, help direct its movement, and often help initiate the process of translation. The third change that happens during RNA processing is the excision of introns and the splicing and retention of exons. So what that means is the exons are kept because those are the ones expressed. The introns are the ones removed. So a memory tool for this, the exons are expressed, so those are what's kept. Alternative splicing is referring to the idea that a single RNA strand can be chopped up and pieced together in different ways. So depending on which exons are spliced together, many different mRNA molecules can be made from a single unprocessed RNA strand. What do you think the function of this is? The function of this is you can increase the diversity of the proteins produced without increasing the amount of DNA you need. So the very same nucleotide sequence in DNA can result in a bunch of different proteins depending on which pieces of the code are used to form that final protein. All right, one more time, what is gene expression? We're now moving into the second step, so let's see if you can do this one more time. It's so core, it's so important that we get this. One more time, what is gene expression? We're now moving into unit 6.4. We just finished 6.3, so let's stop, and one more time, what is gene expression? Gene expression is using the instructions of DNA to code for a strand of RNA that can then be used to code for a protein. The instructions being used in DNA to create the RNA is transcription. That's what we just went over in 6.3. Now we're going to look at how that mRNA can be used as instructions to create a protein, and this is translation. Where in the cell does translation take place? Translation takes place on ribosomes, and there are both free ribosomes as well as ribosomes attached to the rough endoplasmic reticulum. So that brings up the question, what are ribosomes? Ribosomes are composed of two subunits, both of which are comprised of proteins and ribosomal RNA. If you can remember, in addition to messenger RNA, there are several other kinds of RNA that we'll be looking at in this unit. One of those is rRNA, which composes ribosomes, or ribosomal RNA. And ribosomes are the site of translation. So this is where that polypeptide chain is formed using the instructions of an mRNA strand. So let's look at that process. What are the steps of translation? And as a clue, they're the same steps as we saw in transcription. Those steps are initiation, elongation, and termination, or start, grow, and stop. Let's look at initiation. So there are a couple terms that you need to know here. One is anticodon and one is codon. And you might want to look at this diagram to help you out with those terms. An anticodon is a series of 3 nucleotides at the end of a tRNA molecule that base pairs with a complementary codon on mRNA. More on that in a second. A codon is a series of 3 nucleotides that specifies a particular amino acid or termination signal. So the RNA is read in 3 nucleotide chunks. Those 3 nucleotide chunks are called codons. They are matched up with an anticodon of a molecule that carries an amino acid with it. So 3 RNA nucleotides code for 1 amino acid. And when we're talking about the side on the RNA, or on the mRNA, I should say, that's a codon. And when we're talking about the reverse image of that, so sort of the base paired versions, that's the anticodon. And that's on the tRNA. And a tRNA is what carries those amino acids to the ribosome to create our polypeptide chain. So let's look at initiation in more detail. Use this diagram to describe the process. So what's happening in this picture is the ribosome subunits are coming together with the mRNA to form that translational mechanism. A tRNA molecule carries the very first amino acid to the start codon. So remember, the start codon, or any codon, is on the mRNA. It's a series of 3 nucleotides, and those 3 nucleotides on the codon base pair with an anticodon. And the anticodon is part of the molecule of tRNA. And attached to that tRNA is an amino acid. So depending on what the codon is, it will match up with a different amino acid. Next step is elongation. Take a look at this diagram and describe the process. In elongation, amino acids are brought to the mRNA by tRNAs and are added to the growing polypeptide chain. And again, it's that anticodon of each tRNA that base pairs to the codons on the mRNA. So which amino acid it is depends on what nucleotides are in the codon. Last step, termination. What results in the termination of translation? That's a stop codon. So when we get to a particular codon, UAG, that signifies stop. And so instead of connecting with a tRNA molecule, that codon connects with a release factor. So a stop codon in the mRNA is recognized by a protein release factor, and translation stops. This releases the polypeptide chain. If you remember, the amino acids, when they're all being built together into a polymer, is a polypeptide. Since a polypeptide chain is made of a sequence of amino acids, the amino acids need to be in a particular order. So how do we know what order that is going to be? How can you determine the order of amino acids for a given mRNA strand? The way to do this is to keep in mind the idea that each codon on the mRNA corresponds to a particular anticodon, and that anticodon matches up with a particular amino acid. The way we do this is to use one of these tools. We either use a codon chart, you see on the left, or a wheel, which you see on the right. And we take that codon, which has 3 nucleotides of the RNA, and we think, what is the first one, what's the second one, what's the third one, and we use these tools to figure out which amino acid that corresponds to. So let's practice this. What amino acid would be added to the chain next if the mRNA codon is CAU? So we would look for the first base of C, the second base of A, and the third base of U, and this would code for histidine. And you can see that's true on both the codon chart and the wheel. Doesn't matter which one you use, you should be able to use either of them. But that's going to tell us that this is HIS, or histidine, is the next amino acid added. And those big letters you see on the outside of the wheel, that's just a single letter abbreviation of all of the amino acids. And because several of them start with the same letter, sometimes that one letter representation can't be the first letter of the amino acid. All right, so let's put this all together. We're going to do some gene expression practice. First step, is this diagram showing DNA or RNA, and how can you tell? This shows DNA because it contains thymine instead of uracil. It's also double-stranded, which is more common in DNA than RNA. So let's imagine that the blue strand of DNA is going to be used as a template. If that's true, what is the sequence of the RNA strand that's going to be produced? And also on this diagram, would the RNA strand be built from left to right or right to left, and why? On this diagram, the RNA is going to be built from left to right because the blue template strand of DNA is read from 3 to 5, which runs left to right, and the corresponding RNA is built from 5 to 3, which is also left to right. So in this case, the template strand of TAC, TAG, A is going to give us AUG, AUCU, as you can see in the middle there, and then the full transcript is shown below. All right, so here we have our transcript. What's the next step of gene expression in eukaryotes? That's post-transcriptional modifications. Remember, we get a cap, a tail, and exon splicing. We're not going to see that in this practice. We're trying to keep this simple, so let's just assume those already happened. What's the next step? Next step is translation. Reminder that translation occurs at the same time as transcription in prokaryotes and after post-translational modification, post-transcriptional modification in eukaryotes. So let's use a codon chart or a codon wheel to translate this RNA strand. So when we translate this RNA strand, AUG is one of the start codons, and this codes for methionine. AUC codes for isoleucine, and UCG codes for serine. Then UAA is one of our stop codons. So this does not code for an amino acid, this just codes for the stop, for the end, termination. You do not need to memorize the full names of all of the amino acids. The information on that would be provided if you ever need to know that. All right, if you're looking for additional practice, here's another strand. First of all, figure out if this is DNA or RNA so that you know if you need to start by transcribing or if you can jump straight to translation. And then do that. Figure out what the primary protein structure would be from this strand. And here's the answer, the key to that practice. You can see this was DNA. You notice that there was T in there instead of U, so that must make it DNA. And then since there was only one, we had to use that as our template. So we transcribed that into RNA, and then we use that to translate into the polypeptide chain. Hopefully that went well for you, that practice problem. If not, go back, try it again. The more you practice this, the easier it gets. You'll get more comfortable using that codon chart, and we'll do more practice in class. So this is where we'll leave this video off.